All right, good morning, all. It's a pleasure to be here to kick off day two. I'm here with my AMP heart failure colleagues and to tell you about our relatively young AMP in heart failure and heart failure preserved ejection fraction. I mean, it was particularly gratifying to hear all the talks from the more mature AMPs yesterday to learn that we're all fundamentally trying to address a similar problem of taxonomy and to do so with fundamentally similar strategies. In our case, we're dealing with a condition of heart failure with preserved ejection fraction, which is defined by a really poor clinical descriptor, and that's the capacity for the heart to squeeze out its blood content. And that says nothing about mechanism, and it belies a very diverse and heterogeneous population. So very much like the other AMPs that we heard from, our approach is very similar, and that's to collect as much information as we can, both cross-sectionally and longitudinally, and that includes clinical data, environmental data, as much imaging data that we can, multimodal, as well as functional imaging data, functional provocational exercise, and pulmonary mechanics to capture the dynamic aspect of heart failure. And when I say dynamic, I mean beat-by-beat variation in the load upon the heart as it's trying to accommodate to the ever-fluctuating needs of the body and its energetic demands, as well as the panoply of omics data, and molecular descriptors, and then critically tissue. And tissue in this case, in our flavor, is from the heart, from living patients in the spectrum of disease. And with this information to create a digitized, high-quality, harmonized data set that ultimately will be democratized for crowdsourcing, and for our own bespoke and prioritized questions to leverage advances in analytic approaches like network analysis and machine learning to achieve that elusive goal of a stratified population of patients that are defined by their molecular descriptors, that have associated biomarkers to readily identify them, that have distinct clinical trajectories, and for which we, people like me, can create drugs and treat the right patients. So why heart failure? First, it's an incredibly prevalent condition. About 30 million people globally will have heart failure by the end of this decade. 1 in 4 to 1 in 3 will develop heart failure within our lifetimes, which guarantees that virtually everyone in this room will either have or have somebody that they love develop heart failure. And the epidemiology is clear. HFpEF, preserved ejection fraction, is now equal to all cases of heart failure, and it's rapidly eclipsing the more well understood, at least therapeutically treated, heart failure of reduced ejection fraction. And like everything that all of us have been treating or are trying to focus on, heart failure is marked by significant disparities. And this is disparities across for prevalence as well as outcomes based on race, gender, and economic wherewithal. And it's not just the prevalence, it's also highly mortal. Half the people who develop heart failure are going to die from it within 5 years, and that makes no difference what flavor of heart failure that you have. And there is very little in the form of therapeutic options. We've been quite fortunate recently to see the development of the SGLT2 inhibitors, and that they have had benefit across the spectrum of heart failure. But really the benefit in HFpEF is in terms of hospitalization. There's a great impact upon quality of life, and when it comes to mortality, little, if any, benefit. And so why is this such a big problem? I alluded to the heterogeneity, and the heterogeneity is manifest. There's great heterogeneity in the insults that can promote heart failure that are intrinsic to the heart, and many of these are kind of graphically shown here, as well as the comorbidities throughout the body that may impose the burden and the injury upon the heart, these systemic comorbidities. And it's very difficult to know from any particular patient which of these mechanisms are the major driver, and none of these say anything about mechanism themselves. So with this, my job was to set the table, and I'm going to turn to Vandana to show where we're going and why there's reason for hope.
Good morning, everybody. I'm Vandana Sachdev from NHLBI. So Will has nicely laid out the scope of the problem ahead of us. And I'll tell you a little bit more about where we are in terms of phenomapping. This is a term that has been used to categorize patients into distinct subgroups. Will talked about how heterogeneous and complex this syndrome is. And so back in 2015, there was a HFpEF phenomapping study by Dr. Sanjeev Shah, who you'll meet in a couple of minutes. And using the variables on the left of the slide that include clinical data, imaging, EKG lab data, what he did is he demonstrated that he could separate the patients into 3 different clusters, at least 3, and they were mutually exclusive. So since that seminal study, there have been at least 15 or 20 additional studies that basically show the same thing, that you can demonstrate these phenogroups of patients. But when you look across all of these results, we see that there are probably at least 3 predominant phenotypes. They're shown here: the older vascular aging phenotype, a metabolic obese phenotype, and a younger low BNP phenotype. The problem is that there's significant overlap in these groups, and the results are not really clinically actionable. Moreover, the biggest problem is that how these groups map to the molecular signatures within the tissue is not really well known, and it's been difficult to get cardiac tissue. Most of the work has been done with surgical tissue or with donor hearts. So a few years ago, investigators at Hopkins took a very unique approach. In HFpEF patients that were already undergoing a clinically indicated right heart catheterization, they performed endomyocardial biopsies and then did molecular tissue profiling. And what they did initially is they used the transcriptome to demonstrate that, first of all, signatures from HFpEF patients, shown here in purple, are quite distinct from controls and from those with reduced ejection fraction, or HFrEF. Second, in the HFpEF patients themselves, there were unique clusters of subgroups of patients, and when they mapped these patients back to the clinical features, there were distinct clinical outcomes. So these results are pretty exciting. They show that we can stratify HFpEF patients by the molecular signatures, but they're single-center results. The group was about 63 patients in this study. This is definitely something we want to repeat and extend in a larger, diverse cohort. So the same group at Hopkins also performed metabolomics on the myocardial tissue, and even though we use the term normal or preserved ejection fraction, these hearts are actually quite energy-starved. And compared to normals, they are very energy-starved, but they're more similar to HFrEF patients, in fact. More importantly, these are differences that were not detectable by plasma metabolomics. So when we talk about liquid biopsies, in this study at least, the samples were not sufficient from the plasma to differentiate these unique subgroups. So with that, you've heard the importance of tissue for helping us distinguish the mechanistic subtypes. What I'll do in the next minute here is just give you a little bit of an overview of the project design for AMP heart failure. There are 2 parts. One is a retrospective aggregating data that already exists. We're bringing the raw imaging data together, and we have stored biospecimens in many of these studies. And so we can perform new multiomics analyses and try to get to those subclassifications of HFpEF. For the prospective study here in the middle, the core is really creating a new cohort of about 1,000 HFpEF patients that will undergo deep phenotyping. What that means is testing. You'll hear the details shortly, but testing over a course of 2 days. Getting blood, urine, and tissue samples. On top of the deep phenotyping that's in person, there's also a virtual registry in which patients will be answering heart failure-specific questionnaires through an app, as well as EHR data collection. The goal of all of this, as we've talked about, is to get to those different subgroups and the biologic targets for each subgroup so that eventually we can have a master protocol to test precision therapies in these different subtypes. So finally, to give you an idea, excuse me, Will mentioned that we're one of the newest AMPs, so we're quite early. And just to give you a little bit of the timeline, NHLBI awarded 6 clinical sites and a data translation center at the end of 2021. So over the course of a year, the investigators put together a deep phenotyping protocol, and then the AMP was launched at the end of 2022. So we're about a year, maybe a year and a quarter away from that. What's happened since then is that the AMP steering committee has awarded a 7th site. So we were fortunately able to bring in that Hopkins group that's doing the molecular tissue characterization. So we have 7 sites that are doing deep phenotyping, and the AMP has also approved support for the endomyocardial biopsy tissue acquisition and analysis at all of our sites. So what's happened in the last year? Well, we've been preparing for the endomyocardial biopsy protocol. That's a new protocol that you'll hear about. We've been putting in applications for partner data access, harmonizing the extant data, and the DTC has been awarded top med funding for the omics, so that sample preparation is ongoing. And then most importantly, patient enrollment started in the fall of last year, and we're enrolling patients into the virtual registry and the deep phenotyping cohort. So finally, I would be remiss if I didn't acknowledge all of the people that it takes to put this program together. First of all, from NHLBI, Dr. Gibbons and Dr. Goff have been extremely supportive. I want to point to Shweta Ledwa, who's from the NHLBI team. She's not speaking today about Biodata Catalyst, but she's not speaking here, but she'll be in the next panel on portals. We have 2 HeartShare co-chairs that you're going to meet, Swati Shah from Duke and Javed Butler from Baylor. Our Data Translation Center is our central hub, our coordinating center, so you'll meet Dr. Sanjeev Shah shortly. These are all of our clinical center PIs. Maggie Redfield has been leading our tissue working group, and so you'll hear a recorded presentation from her about their activities. Thank you to the FNIH team for getting us through the launch and the first year. Shout out to Harina, our project manager. We do have a treat for you. One of our patients with HFpEF, Cynthia Chauhan, will be giving a recorded presentation, so you'll hear very insightful comments from her. You've met Will already. Ross Upton is one of our private partners from Ultromics, and you'll hear a little bit about his work. Finally, thank you to the FDA. They were very instrumental in our design phase, and HFSA is also one of our strategic partners. So at this point, I'm going to turn it over to each of the speakers. Please come up and introduce yourselves in order. By the end of these presentations, hopefully we've piqued your interest. So if you're not on this slide and you are interested, please come talk to me, Will, or anybody from the FNIH team. And with that, I will close. Thank you, and Sanjeev, you're up next.
Great. Thank you, Vandana. It's really inspiring being here, seeing all that the other amps that have preceded heart failure have done. So today, my job is to talk to you about our data repository and cross-portal integration possibilities. And I think this is really relevant to this room because there are plenty of opportunities for collaboration, as you'll see. Oh, the clicker. I'm taking the mouse. Sorry. Okay. So I thought first I would just, and you'll hear more about this, and Vandana and Will talked about this, but AMP heart failure is our overarching umbrella, and under that is HeartShare. So HeartShare is the program that was set up by the NHLBI to really get this project going, and then AMP heart failure came after that. So when we sort of use these terms interchangeably, but HeartShare is, you'll hear it a lot, is really the program that was just described, and it's really under the umbrella of AMP heart failure. And the HeartShare scientific approach is as follows. We have existing data that you heard Vandana talk about, cardiovascular disease cohorts, HFpEF trials, heart failure with reduced ejection fraction trials, and we have all of that data and the images that we're harmonizing and putting that into Biodata Catalyst, which is where our HeartShare data portal is in the cloud. And from there, we're using 2 approaches. One is a hypothesis-driven approach, because over the years, many investigators have identified pre-existing, predefined HFpEF subtypes, as you heard about, and also molecular pathways that are being leveraged with therapeutics already. And so we can do a hypothesis-driven approach with all this data we've already collected. And then, of course, there's the unbiased approach, so unsupervised machine learning, for example. We then can take these 2 different approaches and look at multi-omics, machine learning, deep learning of images to define novel HFpEF subtypes and molecular pathways or validate pre-existing ones, and then test these and interrogate these even further in the prospective HeartShare component to get to our goal of precision medicine. Again, all of this data is housed in Biodata Catalyst so that it's maximally usable by the investigative community. So what does our HeartShare data repository look like? Well, we have multiple data sources. So one is BioLink. This is NHLBI's platform for all of the NHLBI-funded heart failure trials, cardiovascular trials, some trials of comorbidities like hypertension, diabetes, also all of the cohort studies. And so we have all the data from there, and we're getting the actual raw images from many of those studies as well. There's also additional independent data sets, including industry-sponsored studies. Again, those have data and images, many have omics. There's dbGaP that has its own data and omics from a variety of studies, as you'll see. TopMed is one step up on top of that with all of the multi-omics that's happening and images and data. And then finally, HeartShare. As you heard, there's EHR data, images, there's registry data, and then the deep phenotyping data, images, and omics. All of this, under a massive undertaking, are undergoing de-identification when needed and harmonization, and then they get into Biodata Catalyst. Once there, and of course, it'll be a continual process. We already have some data and images up there. Data requesters could be the HeartShare investigators, could be our AMP heart failure partners, could be external collaborators, or could be AMP collaborators such as yourselves from other AMPs. Request data, and those requests then go to Biodata Catalyst and our Data Translation Center. We then are developing ways to generate cohorts based on those requests. So if we want all patients that have diabetes, for example, or that have Parkinson's disease, we can then do that. Of course, their data in our registries and cohorts and studies, but again, these are not simply cardiovascular disease patients. They have all other kinds of diseases as well. We generate that cohort using an honest broker system so that, that we can keep confidentiality of the patients, and then that goes back to the data requesters, who can then, as I'll show you an example, use this data to further their own therapeutic development if they're, for example, industry partners. So this is a list of all the observational studies and clinical trials. It's really amazing all the resources that are out there that continually investigators have to basically reinvent the wheel every time. Every time we want to do a study that collects and harmonizes all of this data, we have to go through the same process again and again. And with HeartShare, we're hoping to end that and provide one-stop shop for people to come. So for example, all of these observational studies have already been harmonized for dementia, for heart failure, in fact, and we're having to do it again. It's for COPD, for COVID disease research. And the goal really is to do this so that it's there and we don't have to keep going through this. And it's up in a portal, the Biodata Catalyst, that people can access. So thus far, where are we right now? We have harmonized all of the heart failure clinical trial data sets. There are 18 trials. There's 257 variables at baseline and then several others that are longitudinal. And you can see that we have over 27,000 participants. And the majority are men, but in the HFpEF specific studies, it's mostly female. And we have some race ethnic diversity, but something that obviously we're trying to improve over time. And we have a variety of patients across the ejection fraction spectrum. And like I said, there's baseline data and there's longitudinal data. There's cohort studies that have even more, many more variables and number of participants. That harmonization is in progress and hopefully will be done by March. And then next up, we have access to dbGaP dataset harmonization, phenotypes, and omics. So right now, this is just a heart failure clinical trial data set. We really have quite a bit of data, I would say more than many other studies out there, even many meta-analyzed patient-level data studies. So there's a lot for our investigators to work with. And again, as this expands, especially the cohort studies, it'll be even more meaningful to other AMP collaborators across the AMPs. I'm not going to read all of these, but we have a tremendous amount of data that we have gathered and have approval for harmonizing across omics in dbGaP, the database for genotypes and phenotypes. So this, I think, will be really exciting as we try to use the especially whole genome sequencing and GWAS data to then provide a way of validating signals that we see, for example, in the proteome and metabolome. Like Vandana said, we were successful in getting funding of an XO1 application, TopMed, that's NHLBI's program to do multi-omics. And so we have this study that we call MAP-HF, Multi-Omics to Advance Precision Medicine in Heart Failure. And you can see that there are a number of randomized clinical trials and cohort studies that we have biospecimens, blood samples from, where we're going to be able to do whole genome sequencing, DNA methylomics, proteomics, metabolomics, and transcriptomics of the peripheral blood mononuclear cells. You can see that for HFpEF alone, we have over 3,000, and we think we can really leverage this data to find new discoveries. Now, Every one of these studies has already been done, and we already have the biospecimens except for heart share at the very bottom. So in this calendar year, we're going to get a lot of this data on these cohort studies and trials that have already been done, which will be really exciting. Now, besides all of this retrospective extant data sets, we're actually going to our 7 clinical centers, and we're getting EHR, data and images. And this is using an OMOP, common data element model, and we're getting increasingly more sophisticated data from the sites. And we thought we would have 100,000 heart failure patients and 100,000 controls, when in fact we have on the order of 250,000 just from 2017 onwards. And not only are we getting the data, we're getting images. That's our hope. Now, these images may reside at the sites, and we'll be using federated machine learning to access them, but ECGs, echocardiograms, CT scans, MRIs, even pathology images can be used. This is our deep phenotyping cohort baseline visit. You can see that on the right, there's multiple domains where we're going to be getting data from these patients. And the prospective study flow, we're using cutting-edge tools like the Ambra and Flywheel, Microsoft Azure, trying to get all the data and images up there and all linked for people like you, other investigators, other AMPs to be able to use. And so how are we using this? This is just an example where we are now able to take images and run an autoencoder on them, a deep learning framework, where we can take these videos and convert them into data points. We can then aggregate those and cluster those data points and do the same with our omics and come up with HFpEF subphenotypes. And no longer is deep learning really a black box. We can really actually see where the deep learning model is focusing. As you can see here in the example, this was a deep learning model to predict age, biological age of the heart. And sure enough, where the machine is focusing is on the mitral annulus. The mitral valve gets calcified with aging. So it finds something that we already knew about, but think of all the things that we're going to find out that we don't yet know. So finally, I'm going to end with a case study of immunometabolic HFpEF. This is one of those HFpEF subtypes where comorbidities cause systemic inflammation and then cause multi-organ endothelial dysfunction, including the heart. Well, one of the things that we're studying is myeloperoxidase with one of the sponsors of a clinical trial. Myeloperoxidase is expressed by neutrophils and gets trapped in the vascular wall and can cause vascular smooth muscle cell abnormalities, microvascular dysfunction, cardiomyocyte hypertrophy, and cardiac fibrosis. And so we did a small phase 2A trial with an inhibitor of this MPO, and it results in a dramatic reduction of the circulating MPO levels and some hints to potential clinical benefit. But here's where we can use the resource. Now, this was not using our resource, but it's an example where we can interrogate all of the existing studies. We have all this proteomic and genomic data, and we can look at signatures associated with poor outcomes, poor symptoms, poor exercise capacity, as you've seen in this. These are the pathways in orange on the right that are associated. These are the proteins in the circulating blood that are associated with bad outcomes in HFpEF patients. And now we can go back to the trial and say, what does the drug do to these specific proteins? They all go in the opposite direction. And in this way, industry, other collaborators can validate targets and say, okay, we think that this is doing something to the blood that's reversing the process and may provide validation for future therapeutic targets. So here are the take-home points. We're building a massive data repository and really a rich resource that includes images as well. And although these images and omics and data are from epidemiologic cohorts and trials from the cardiovascular disease space, they really have a broad application to all diseases. HFpEF is a systemic multi-organ reserve dysfunction syndrome, and one that actually we think that non-cardiac specific drugs are just as likely to improve as cardiac specific drugs, as we've seen so far. So a real opportunity for all of us. And we think that our Biodata Catalyst platform is really the new cutting edge for AI and machine learning studies. Thank you.
My name's Ross Upton. I'm going to talk about an echo-based AI diagnostic for HFpEF that we've developed. We were interested in developing a diagnostic for HFpEF for many reasons, but as Will mentioned earlier in his presentation, HFpEF constitutes greater than 50% of all heart failure, and it's heart failure's leading cost of hospitalization in the US, projected to cost nearly $69 billion by 2030. One of the reasons why HFpEF causes many hospitalizations is both treating but also the diagnosis remains very challenging. There's often an unclear patient presentation, and that leads up to about 64% of patients being missed with HFpEF in the community. So we sought to develop a solution, a diagnostic solution for HFpEF. This is what the echocardiography pipeline looks like as we think of it. So when you conduct an echo, you have your hardware, which is both cart-based, but also there's new handheld devices. You can plug an ultrasound into your mobile phone now, and so it's a very scalable technology. You then have guidance systems, which help untrained users acquire very good echocardiography images, and you have places to store those images called packs. And then we take over from the next phase, which is how do you sort those images? So we use AI to label the images within the packs, produce automated measurements, and we're the first to bring through a diagnostic for echocardiography, and in this case it's HFpEF. This is what our pipeline looks like that goes into the diagnostic. So you have a 3D neural network model to identify the views, and in this case we were interested in identifying the apical forechamber. You have an auto-contouring engine that contours the left ventricle to give us measurements. You have a machine learning model to select the right heart cycle, and then that produces an ejection fraction, and if that's preserved, if it's greater than 50%, it then feeds through to our 3D neural network model that works directly on the pixels to detect HFpEF. And so this is the product, this is what we developed. It works off a single apical forechamber view and outputs a diagnostic recommendation of whether a patient has HFpEF or not, and it also outputs a probability from 0 to 1 as to where the patient is on that scale. We designed it to work just on an apical forechamber because we wanted it to be as scalable as possible. You can imagine there's probably 100 echo images taken per patient, and if we required all 100, it wouldn't be that scalable. So we wanted to see if we could do it off just a single image, and that way it could make its way into the community rather than restricted to expert hospitals. We built it in partnership with the Mayo Clinic and Dr. Patricia Pellicer. We used nearly 7,000 patients to train the model. And we used the definition of the ejection fraction had to be greater than 50%. They had to have a clinical diagnosis of heart failure, and there had to be increased filling pressures, either present on the transthoracic echo or by right heart catheterization. And these were the results. So we tested it in an independent population of 1,300 patients across 8 sites, across 6 different states in the US. And we were able to achieve a sensitivity of 88% and specificity of 82%, and the accuracy was 91% in that population. And we compared it to the current clinical risk scores that are used, both the HFA and H2FpEF score, to see how it performed in that cohort. And we saw in the 1,300 patients, both the HFA and the H2FpEF scores yielded a large amount of indeterminate diagnoses, 60% for H2FpEF and 54.5% for HFA. And our model was able to rebucket these indeterminate patients into both diagnostic positive and diagnostic negative cases. And in comparison, we only had a 7% non-diagnostic rate. So this is one of the real challenges with HFpEF, is diastology is often indeterminate, and this is why it's difficult to pick up these patients. We then looked to see how did it fare over 5-year outcomes. We had 5-year outcomes on all of these patients. And if we diagnosed you as positive, there was a 90% higher probability of dying over 5 years versus diagnostic negative. Even more than that, we've now looked into the probabilities. If you were above 0.75 on our probability versus below 0.25, you had a 17-fold increase in cardiac mortality. And this was published in JACC Advances a few months ago. Next, we looked to develop further modules in this space, and amyloidosis was the obvious next step for us. Amyloidosis is the big masquerader of HFpEF, as Sanjeev would call it. It often looks like HFpEF and causes heart failure. However, it's very important to isolate amyloidosis patients as they have a very different treatment pathway. And in fact, if you do end up treating them for heart failure, that can be detrimental to the patient. So it's really important to isolate this phenotype. We actually think a big reason why a lot of the HFpEF clinical trials in the past have failed is because there's been undiagnosed amyloidosis within those cohorts. A little bit about how we deploy the model to hospitals. We have a secure VPN connection into the hospital PAC system, so the echoes are taken in the normal way. They're stored in the PAC system, and we have a VPN. We absorb the images into our cloud and then turn around a report in about 15 minutes after it goes through the AI pipeline. There's two ways we can receive the images. Either the clinician decides, I want the echo go report on that patient, and right-clicks and sends it to us, or we have an automated send based on patient indications. And for the first time ever, we have 2 reimbursement codes for a non-invasive diagnostic for HFpEF, both in inpatient and outpatient, and these are very new. The outpatient one just came in January of this year, and the inpatient one came in towards the end of last year. And so this will enable more clinicians access to our technology as well, which is very exciting. And lastly, we obviously have partnered with the FNIH and joined the AMP. And I think for us, the reasons are we felt our technology could help with the trial. There's a lot of synergies with how the program was designed with what we were working on internally. And so we think the technology can help with detecting HFpEF, can help with the phenotyping of HFpEF, and can help find HFpEF patients if sites are struggling for recruitment. We can help find more HFpEF patients. And then our amyloidosis algorithm can help isolate perhaps undiagnosed amyloidosis within the cohorts. And then what we get out of it is, one of the main things so far is unparalleled access to experts, such as my esteemed colleagues on this panel. They get to stress test our technology, get to give us feedback about how it fits in the clinical workflow. And as we design the future for how HFpEF is supposed to be diagnosed, we can see how our technology fits into that pathway. And for us, that's incredibly valuable, and that's sort of unparalleled access that we wouldn't get elsewhere. And that's it. Thank you. And now I think we're going to hear from Dr. Maggie Redfield at the Mayo Clinic in a pre-recorded presentation.
Hi, I'm Maggie Redfield, and along with Dr. Barry Borlaug, I co-chair the Mayo HeartShare Center. And I've also been working as the co-chair of the HeartShare Cardiac Tissue Working Group. And I'm here today to tell you about 2 very exciting new studies that we're starting in HeartShare. The first is called Myocardial Mechanisms in HFpEF. And in this study, we will do multi-omics in cardiac tissue from HFpEF patients and controls. Tissue will be prospectively obtained via research endomyocardial biopsy in HFpEF patients, and the tissue from the controls will be provided by the University of Pennsylvania Cardiac Tissue Repository, and will include both patients with HFrEF and patients without heart failure. This study has been approved by the Heart Failure Observational, or the HeartShare Observational Study Monitoring Board, and it has been funded by the AMP. And protocol startup at all the sites is, all but one is underway. And the second study I'll tell you about is called Myocardial Mechanisms in HFpEF, Insight from Historical Endomyocardial Biopsies. And here we have an active protocol draft that's been submitted at Mayo, and we're doing pilot studies to prove feasibility and finalize study design. I would like to acknowledge the HeartShare Cardiac Tissue Working Group, and they have really developed these protocols and will help implement them. And I'd like to especially acknowledge Jason Duran, who is the co-chair, And the fact that all 7 sites from HeartShare have actively participated in this working group, and also acknowledge the overall HeartShare leadership who have been very involved in the working group. So why heart tissue? Well, because even though renal dysfunction and skeletal muscle abnormalities and adipose tissue and pulmonary vascular remodeling can influence the HFpEF phenotype, at the end of the day, the fundamental underlying problem is abnormal cardiac structure and function. And if we understand the cardiac pathophysiology driving these changes, we can identify new drug targets. And animal models can help in this. However, they may not really replicate human pathophysiology. So we really need to study human HFpEF myocardium. So to kind of start this process, we wanted to understand what do we actually know from looking at human cardiac tissue from HFpEF. And so we did a scoping review. And even though there's been over 20,000 papers about HFpEF, there's really only 56 that describe studies in human tissue. And these were mostly published in the last 10 years and in actually very good journals. And they used tissue from a variety of sources. Some were patients undergoing surgery for different cardiac conditions. Some were patients having RV endomyocardial biopsies, and some were autopsy studies. And really these studies consistently described variable severities of abnormal cardiac structure and function, i.e., increases in myocardial fibrosis, cardiomyocyte hypertrophy, microvascular rarefaction, T-tubule dilatation and proliferation, impaired calcium cycling, and hypophosphorylation of the giant molecular spring titin. And studies showed impaired metabolism with lower mitochondrial area and fragmentation, cristae destruction, and degeneration of the mitochondria. And these studies also showed impaired fatty acid oxidation without apparent use of other alternative fuels. And some of the studies suggested that this could be associated or due to NAD deficiency. And these structural changes were associated with impaired relaxation and increased passive force or myocyte stiffness. So pretty good understanding of structure and function, but not very much data on the fundamental underlying pathophysiologic biochemical pathways that were mediating these changes. And really only about 8 pathways have been studied in human HFpEF myocardium, and only 4 pathways was there any evidence that they might be involved in HFpEF. And for 2 of these, it was just 1 study suggesting that. So we really need a much better understanding of the underlying pathophysiologic changes that are going on. Now, all these studies were hypothesis-driven. So they have these precious cardiac samples, but they only really looked at 1 pathway. And now we have, with omics, obviously, the capacity to look at all pathways, basically, at once. And we know that we can do genomics, transcriptomics, proteomics, metabolomics, and in some cases, cytomics. And we've learned thus far that it's really important to do, at a minimum, at least transcriptomics and proteomics, because sometimes you need both to understand the direction of the pathway. So that's really, really important. And in our scoping review, really, there were only 2 studies of transcriptomics in human HFpEF myocardium and 2 studies of proteomics. And these were all pretty small studies, and they had very different findings. For instance, with the transcriptomic studies, both studies had pretty good coverage, showing about 13,000 genes were detected. But in one study, 60% of these genes were differentially expressed in HFpEF versus non-failing controls, and in the other study, only 5%. So we really need a lot more understanding. So that is the first study, and we hope to get started very soon. And while we're gathering these samples, we're going to be working on this next study. And this really uses endomyocardial biopsies that we have on hand. So when you see a patient with HFpEF, you need to rule out certain specific causes that have specific treatment. And that might be a genetic cardiomyopathy, an infiltrative process, constrictive versus restrictive physiology and underlying myocardial findings, or inflammatory cardiomyopathy. And now we can pretty much do this in most patients with imaging, but in the not too distant past, we had to do endomyocardial biopsies to determine that these specific types of cardiomyopathies were present or not. So when you do these biopsies, you take between 3 and 6 samples, and you want to have nice big samples. You don't always get as big a samples as you want, and they're all placed, formalin fixed and placed in one paraffin embedded block. So even if you exclude cardiac transplant patients, which is the most common cause for the need to do endomyocardial biopsies, if you exclude them, then you have all these different phenotypes of patients. And each site has taken a quick look and has from tens to actually thousands of historical endomyocardial biopsies. And they're all formalin fixed, paraffin embedded, and include patients with different phenotypes of HFpEF, HFrEF, and a smaller number that could really serve as controls. Now, they're formalin fixed and paraffin embedded, and that limits to some degree what you can do with them, but not as much as it used to. And really, we are starting some pilot studies to characterize these patients, and then pilots with tissue microarrays, digital histopathology, and spatial transcriptomics and proteomics, and possibly even mass spec proteomics. So this will be something we can do relatively soon and can be very helpful. So we're very excited about these 2 studies, and I hope that you will be too, and I appreciate your attention today. Thank you.
Thank you for inviting me to speak with you today. I am Cynthia Chohan, and I have stage 3 HFpEF with significant comorbidities, including but not limited to stage 4 kidney disease and osteoporosis. The good news is I am celebrating 9 years of surviving with HFpEF, and in March it will be 10 years, which, as you know, puts me in a class of rare people with a common, life-threatening, life-limiting, life-altering disease. I think today I want to tell you first that I am so grateful for all of you and for how deeply you care about those of us who live with HFpEF. The other part of my good fortune is that I am cared for by an outstanding specialist, Barry Borlaug, and that I am fortunate enough to go into clinical trials to help understand, treat, and hopefully someday cure this debilitating disease. As you know, the majority of people with HFpEF are older women, so I would like to ask you to change your usual presentation slides where your illustration is of a man. I think that is misleading and disrespects women. I would like for you to have your slide pictures illustrate those who are in the majority, older women. It is already hard enough for us to get an accurate diagnosis and care. Be sure nothing in how you present fosters that problem. I'd like to start with Emily Dickinson's Hope is the Thing with Feathers. Hope is the thing with feathers that perches in the soul and sings the tune without the words and never stops at all. And sweetest in the gale is heard, and sore must be the storm that could abash the little bird that kept so many warm. I've heard it in the chillest land and on the strangest sea, yet never in extremity it asked a crumb of me. And so I am living hopefully, and I think successfully, with long-term HFpEF. What does that mean to me as a patient? Before I share that with you, I want to be very clear that I am not complaining. If I live the rest of my life where I am functionally right now, that is okay with me. Rather, I simply hope to help you understand the daily life of a person with HFpEF and comorbidities. More than anything, I'm grateful to be alive and functional. What I'm going to share is simply the price I pay for that honor. Ironically, over the course of years, I have lost 5.5 inches of height. Even though my weight is 190, if I still had my original height, I would be simply overweight. But because I've lost height, I am obese. As a person who has struggled with obesity all my life, that hurts. Then there are the usual things that you know: low sodium diet, exercise, appropriate monitoring, and balancing the demands of multiple serious comorbidities. What does that look like in my daily life? My life is severely attenuated by this condition and its comorbidities. I know you understand the medical aspects of this collusion. I want to talk with you about the life experience of it. The fatigue is ever present. I'm sure my heart failure contributes to the fatigue, but I'm guessing that my kidney failure and severe arthritis do their part too. For example, I have finally accepted that I am deconditioned. And while it is easy to say that's because of the heart failure, in reality, I think it is because the severe arthritis makes movement very painful. So I am inclined to be still. And when I am still, I breathe better. And I don't hurt as much. Medication that was helping control the arthritis pain and promoting movement had to be discontinued because it was also promoting my osteoporosis. Other pain control medications used to treat arthritis are detrimental to my heart and kidney. I am much slower in my daily life. I can't do a lot of things that I used to do easily. I joke that if you ever see me in a race with a snail, bet on the snail. So I use humor to avoid feeling devastated. I've become more dependent on the help of others, which is very difficult because I was always a fiercely independent person. People who love me tease me that I am irrationally independent. Both loved ones and strangers are kind and helpful, but needing that help is difficult for me. Breathing is really hard, especially when I try to do anything other than sit quietly. I'm sure that you are aware that the cadence of my speech while I talk to you is stilted at best, and that's related to my breathing problems. I know some of my issues are complicated by the comorbidities, but knowing that doesn't make things easier. I have to sleep with my head significantly elevated and my feet elevated. So when you imagine me sleeping, think of a badly arranged, obtuse triangle. I'm very good about my diet, but eating isn't very much fun anymore. Mrs. Dash and spices only go so far to replacing salt. I guess you could say having HFpEF has helped my math skills because I spend a lot of energy making sure I measure everything I drink and everything I eat, as well as balancing what I can eat for the health of my heart versus what I can eat for the health of my kidney versus what I can eat for the health of my bones, and so on. On the other side of what I can eat are all the things I can't eat. I don't only eschew salt. I also eschew high potassium fruits and vegetables. I have to eat some animal protein to fight muscle wasting, but I can't eat too much to protect my kidney, and I can't forget getting enough calcium to try to forestall my osteoporosis. Don't forget the trials and traumas of deconditioning. There's the issue of having enough energy to prepare interesting meals. But before any of that, I have to get up in the morning. I consider it a triumph that I can still get up, make my bed, and catch my breath fairly quickly. Getting dressed has become something of an Olympic exercise because the movements of dressing are quite painful and they take my breath away. Small victories define my life now. And of all the small victories, staying alive counts really highly. It's not just about staying alive, about about existing, but also about having a good quality of life, seeking the balance between quantity and quality. I can no longer walk briskly, much less run the 3 miles I used to run, but I can walk in my garden and slowly down my street. Small victories. Every day is a challenge, but I still believe that every day is a gift, and I am grateful that I am still alive and fairly functional and can enjoy that gift. One question on a survey that I do regularly regards whether or not I would be content to stay at the level of function that I am now. The answer to that is a resounding yes. I choose to focus on what I have, not on what I don't have. Focusing on how little treatment and intervention opportunity there is for HFpEF does not enhance my life. I can grieve about how hard it is to breathe, or I can be grateful that I can still breathe. I choose gratitude. If I cry to the universe, why me? The only possible answer the universe can give is why not? So, why am I sharing all of this with you? Certainly not to bring down your day with my issues. I am sharing it to emphasize how deeply important I think the work you do is in the HeartShare study. Deep phenotyping gives me hope that there are important undiscovered ways to intervene in the course of the systemic problem. Through deep phenotyping, you can better understand why HFpEF happens, how to forestall its development, and possibly even how to eliminate its development. Elucidation can lead to elimination. And so I am in the HeartShare trial to help move us closer to treatment, cure, and extinction of HFpEF. I encourage you to invite as many of your HFpEF patients as possible to join this journey of discovery. I want to close by saying again, thank you for all you do, for your investment in and your commitment to helping those of us with HFpEF live longer and better. You are truly a gift in my life and in the lives of all the other patients whom you see. And so I give you my greatest and fondest thanks.
I'm Javed Butler. I'm a cardiologist and a clinical trialist in Dallas, and along with Dr. Swati Shah, we will give you a short presentation. You heard from our colleagues how this program started and what the plans are, so we'll just give you a little bit of an update on where we are today and where the program is headed moving forward. I have to say, though, the patient representative that we just heard from, Cynthia Chauhan, I have had an opportunity to know her over the past several years. And I can categorically say that every time I've heard her talk, she has sort of re-inspired me of the things that we are doing and that why it is so important for us to do it very enthusiastically. So appreciate the patient's perspective into all of this. So this program was jump-started by a foundational investment by the NHLBI for $25 million for the AMP program. There were 2 RFAs out, one seeking several clinical centers to enroll patients and one data translational centers. Through a competitive process and the review process, these were the sites that were chosen. Dr. Sanjeev Shah and Northwestern University was chosen to lead the data translational centers. The 6 clinical sites that were chosen for the HeartShare program was Mayo Clinic, UPenn, UC Davis, Wake Forest University, Northwestern University, and Mass General Hospital, Brigham and Women's Hospital as well. I was particularly honored that Dr. Swati Shah at Duke University and myself were asked to co-chair the program. As you heard the importance of myocardial biopsy in understanding the true pathobiology of HFpEF, it became very evident that that would be a major focus of the program, and the idea was to then seek another center that has a specific expertise and focus on endomyocardial biopsy. So after going through another round of solicitations, Johns Hopkins University was also chosen to be added as a seventh site, and that site with specific expertise on myocardial biopsies, not from their own site as well, but also to help us diffuse their protocols and how they do it across the other clinical centers as well. So as we saw this sort of the funnel that Dr. Vandana Sachdev showed, that the data collection occurs at sort of three different levels. So one at the site level at the top is to collect a contemporary data of all the patients that are there within all of these clinical sites to understand real-life epidemiology of patients with heart failure and preserved ejection fraction. From that larger pool, invite some of the patients to join in a prospective low-touch registry in which there won't be any particular face-to-face contact or any procedures, but these patients will be consenting for us to contact them, get some information in terms of some questionnaires, and have a longitudinal follow-up a little bit more deeper than what might be there in the electronic health record by itself. And from that, those patients that are motivated to then go into the deep phenotyping cohort will be invited to go through the rest of the procedures. So this is sort of the platform that has been set up for the study. The patients who will enter the deep phenotyping protocol will really go through deep phenotyping. So this will include cardiac structure and function. There will be an exercise echocardiography coupled with exercise testing, cardiac MRI, lung structure and function, PFTs, lung CT, body composition with CT, vascular studies with aortic stiffness and pulse wave velocity, frailty assessment, and a whole array of biomarker and omics as well. Also, in a subset of the patients, they will undergo skeletal muscle biopsy, they will undergo endomyocardial biopsy, and also fat biopsy as well. So where are we today? So I was remiss to say, so where are we today is that in this big registry of all the patients, there are about 50,000 patients or so per site. That data collection and aggregation is in process right now. About 56% of those patients in this real world heart failure, what is happening in all of these sites, actually validates what Dr. Chaktaw originally mentioned, that the epidemiology of HFpEF has actually taken over HFrEF. So about 56% of the cohort is patients with HFpEF. Of these, about 675 patients have now enrolled in a virtual registry, and of those, about 65 patients have enrolled in the deep phenotyping protocol. So the study is well underway. What are some of the characteristics of the patients that have been enrolled? So in the larger registry, it's about 50-50 male and female, and about 82% are non-Hispanic white population, and about 7% of the black population, and a minority of the other race and ethnic groups. In a specific smaller cohort of the deep phenotyping patients, there is about 61% female participation and about 39% male population, which is more consistent with the disease distribution in the real world as well. Also, the distribution of the race and ethnicity is somewhat parallel to what we are seeing in the larger registry. The sites are using various different methods to increase the enrollment into the study. Some of these are interventions that are based on the site clinics. Some of them are based on leveraging the electronic health record and other electronic means to get in touch with the patients. Some of them are trying to make it easier for the patients to participate, and obviously using personal contacts with the participants. Now, it is really important aim for the project to have a well-diversified population that is enrolled in the study that mirrors not only the population at large, but specifically the population that is afflicted by the disease. So these are some of the targets that we have set for ourselves of what we want to achieve in terms of diversity of the patient population, including about 20% of the black population with HFpEF and about 10% Hispanic or Latino population. In order to achieve the diversity goal, there have been partnerships that are developing at the site with various different faith-based group, community health program, community health workers, other health equity programs, and not only trying to reach for a diverse patient population, but also create a diverse recruitment team that can relate to those populations and also aid in the recruitment of the patients. And at this point, I will hand over to the more intellectual part of the presentation to my colleague, Dr. Swati Shah.
So I quickly just wanted to highlight what an incredible set of sites we have and some expertise across the sites as we delve into key research questions in this overall paradigm of trying to dissect the heterogeneity of HFpEF and identify HFpEF clinical, physiologic, imaging, and molecular sub- and endophenotypes. We have incredible expertise across our sites in innovative image-based researching. You know, for many years, we've had pretty crude techniques for diagnosing HFpEF, which are very nonspecific, and specifically with echocardiography. Our colleagues, our site at Northwestern has expertise in four-dimensional flow MRI. This is able to look at in vivo hemodynamics and regional flow variation, velocity, and volumes. Our colleagues at our site at the MGH Brigham is using artificial intelligence to look at pulmonary vascular and arterial anatomy as well as tortuosity, which using these AI algorithms is associated with different types of pulmonary hypertension in patients with heart failure. And finally, UC Davis site has a really cool expertise in whole body PET, really comprehensive whole body evaluations of inflammation as well as looking at inner organ communication. And the idea here is really to use this imaging to identify not only these HFpEF subphenotypes, layering that molecular data on top of those subphenotypes for more targeted precision therapies, as well as improved diagnostics and prognostics for HFpEF. I want to give a huge shout out to the mitochondria, the powerhouses of the heart. 1/3 of the cellular volume of the cardiomyocyte is mitochondria. We have a lot of expertise in evaluating mitochondrial function. Our colleagues at Wake Forest have Seahorse and other assays to be able to look at mitochondrial capacity and especially at the role of skeletal muscle in HFpEF. I also wanted to highlight work from our new Hopkins site on the right. Dr. Sachdev also referred to this particular study where they did mitochondrial metabolism and looked at fuel substrate metabolites in the myocardium of HFpEF and found several different pathways that were dysregulated. And in fact, we've seen fatty acid oxidation defects in the blood, but when you look in the heart, those defects that we see in the blood are actually not coming from the heart, based on our Hopkins colleagues' studies where there was really a disconnect between the blood and the myocardium, suggesting that the pool of skeletal muscle, which is actually our larger sort of pool of source of fuel substrate metabolites, may be accounting for this. And so really in our HeartShare AMP heart failure, we'll be able to evaluate this inner organ communication between the heart, the skeletal muscle, and the adipose tissue as we think about mitochondrial metabolism and fuel substrate utilization. Lots of work being done to identify new molecular and cellular pathways using evolving technologies for large-scale proteomics in the blood. We've identified circulating proteins of known pathways, but also novel pathways. Our colleagues at Hopkins have also done RNA sequencing, bulk RNA sequencing in myocardium from HFpEF, as Vandana referred to, identifying known pathways, but also novel pathways. And importantly, all this work is really in the myocardium, mostly has been done in bulk, right? But when you look at this UMOP plot down at the bottom left, which is actually in HFrEF myocardium, we know that there's marked cellular heterogeneity in the myocardium. So we really need to look at these mechanisms at a cellular level, and the fact that we'll be able to get tissue, myocardium, and skeletal muscle and apply evolving single cell and single nuclei transcriptomics and proteomics will help us understand these pathways at a cellular level. You've already heard a lot about our expertise, both from our Ultromics colleagues as well as our sites, using artificial intelligence and machine learning approaches, including the machine learning phenomapping. Mayo Clinic has expertise in ECG algorithms that are very correlated with diastolic dysfunction, which is a key component of HFpEF. I already told you about the AI work that's being done at the Mass General Brigham for pulmonary vasculature and heart interactions, and expertise at Mayo Clinic in digital microscopy. And then finally, I just want to point out physiology is really important. I know that seems sort of self-evident as we think about the heart, but this is really deep integrative physiology. We have expertise across our sites in this space. This is not only important as we think about sort of what we already know about physiology and HFpEF, but as we think about evoked physiologic phenotypes. A lot of patients actually won't have symptoms until they have exercise or some other physiologic insult. So our ability in HeartShare and AMP heart failure to evaluate deep physiologic phenotypes and look at multi-organ and systemic reserve capacity, including things like chronotopic incompetence, peripheral oxygen utilization, and RV, pulmonary vasculature, systemic reserve capacity, and looking at inter-organ communication in the context of that. Physiology is really, I think, a unique aspect of this study. So I just want to compliment our sites on the incredible group of people who have come together.
So, Courtney, I'm getting the nod from you that we've probably run over time. Is that the case? Okay. So then on behalf of all of us, I want to thank you all for your attention and the privilege of being here to share this AMP as part of the larger AMP Symposium. Thank you.